So this is all about Chapter 11 on Mendelian inheritance. So essentially, way back in the day, Gregor Mendel is an Australian monk who's given the term the father of genetics. So basically, he studied science and mathematics at the University of Vienna, and his research was exclusively on the garden pea plant, and that is what gave us all of our knowledge about inheritance. And the craziest thing about Mendel was that he was able to formulate the laws of heredity in the early 1860s with absolutely no knowledge of cells or chromosomes. He didn't even have a microscope, which makes me feel so terrible because I haven't formulated the laws of anything and I have access to all kinds of technology. The reason why Mendel's information and why his work was so believable was essentially that he focused his energy on completing these experiments. He did many experiments, and his results were always mathematical data. And so it was really hard for anybody to debate his mathematical data that he was consistently getting. So essentially some review. Make sure that you note that a gene is a portion of DNA found on a very specific region of a chromosome that has the information that is basically the information to create the correct protein. And then remember that a protein always comes out as an expressed trait. And some other important things to note is that an allele is a form of a gene that has just a slightly different nucleotide sequence. So like we all have the same genes or DNA segments on our chromosomes for eye color, but we have different alleles or variations of that. So that causes us to have different variations in eye color. So Mendel used the pea plant, which was very good for use in his experiments because it was really easy to cultivate. They had short generations. Normally they could be self-pollinating, but they could be cross-pollinated by hand as well. You could find true breeding varieties available, and the traits were simple and objective. So if you look, there's some of the traits that were important for Mendel. It shows you like stem length, pod shape, seed shape. There's all kinds of traits that were super easy to work with. Back in the day, inheritance was thought of as a blended inheritance. So the thought was if you had a tall parent and a short parent, they'd make a medium-sized kid. But Mendel's data figured out that that was completely different. And so because of that, Mendel was able to formulate the particulate theory of inheritance, which just means that inheritance involves reshuffling genes from one generation to the next. Essentially, Mendel, what he did was he performed some crossbreeding experiments. At first, he used true breeding homozygous plants, and initially he started with looking at plants that differed in only one trait. So like a tall plant and a short plant, and everything else would be the same. He did some reciprocal crosses. He looked at the parental generation, he looked at the first generation, which he called the F1 generation, and he looked at the second generation, which he called F2. And because of his data with monohybrid crosses, he was able to formulate the law of segregation. So essentially, what he figured out was that each individual has a pair of factors for each trait, and those factors separate during gamete formation. And then when that happens, each gamete has one factor from each pair of factors. And then fertilization brings those factors back together. So the pea plants were controlled by two alleles. If it has a dominant allele, it's expressed with a capital letter. If it's recessive, it's a lowercase letter. And every time there's a dominant or capital letter present, it's always going to mask the recessive trait. And so alleles can be found on homologous chromosomes at a particular gene location, and they can be homozygous, which would be identical, or they could be heterozygous, which would be different. So if you look at the classical view of homologs, You'll see like big G, little g, that would be heterozygous. But if they are both capital G or lowercase g's, those are homozygous. Also, make sure you pay attention to the terms genotype and phenotype. So genotype refers to what the genes look like, and phenotype is basically the physical appearance. This is an example of a monohybrid cross done by Mendel. So he looked at a tall plant and a short plant, and he got the gametes from that generation. He joined them together, and he would get a heterozygous offspring. Then he would look at the gametes from that heterozygous offspring, and he would cross those, and every time he did that, he would get 3 tall ones and a short one. So every time he did this monohybrid cross with that and looked at that second generation, that F2 generation, he would get a 3-to-1 ratio. It's pretty hard to deny data that has a 3-to-1 ratio in every trial you do. And look at the numbers that we're working with. So we're not talking like, oh, Mendel did 5 trials, so he's good. He did hundreds upon hundreds, even thousands of trials per data point. And every time he did it, he got a 3-to-1 ratio, which is a pretty big deal, really. He didn't stop there, though. Then he looked at a dihybrid cross. So this is when he looked at plants differing in 2 traits. And so he tracked those traits through 2 generations. And because of that information that he got from dihybrid crosses, he was able to formulate the law of independent assortment, which means that the pair of factors for one trait separate independently of the factor for other traits. And so because of that, all possible combinations of traits can happen. And we know this is due to the divisions in meiosis. We know that is a fact. This is what a dihybrid cross done by Mendel would look like. So we have the parental generation that's homozygous for the traits. Then we take that second generation and we look at all possible gamete creations and then all possible outcomes. And every time he did this, just like when we practiced in class, he got a 9 to 3 to 3-to-1 ratio. So we use Punnett squares a lot in lab, and it's a really good way to look at what might the potential offspring look like. And so if we're looking at this particular parent, they're heterozygous for a trait, and they give you a 3-to-1 ratio. Sometimes a genetic disorder can occur. So that's basically a medical condition caused by alleles inherited from parents. It can happen on an autosome, or it can happen on a sex chromosome. So either way, an autosome is basically any of those chromosomes, number 1 through 22, that happen that you can see on a karyotype, and then you have the X and the Y, or the XX. So genetic disorders that happen on autosomes are called autosomal disorders. Some of those disorders are dominant, some of those are recessive. If it's autosomal dominant, an individual with capital A or capital A, little a, they do have the disorder. But if it was little a, they would not have the disorder. So an autosomal recessive disorder, if an individual has an autosomal recessive disorder, capital A or capital A, little a, they do not have the disorder. If it is heterozygous, they wouldn't have the disorder, but they would be a carrier. So an individual with little a, they would have the disorder. So this shows you an individual, and it shows you what's called a pedigree. So pedigrees are really good ways to look at lineages, and really good ways to look at what an individual might be inheriting. So this would be like the grandparent, grandma, this would be grandpa. Right here is their offspring. So they had 2 daughters, right? And then they have, this is their person, this is their person, and then they have 3 children, they have 1 child. Now, it does show you a double line here, and that's where Cousin Bill found Cousin Susan, and they had some offspring. So, with autosomal recessive disorders, most affected children have unaffected parents. Heterozygotes can have the unaffected phenotype because it's autosomal recessive. 2 affected parents will always have affected children. They can only give little a's in that case. And close relatives who reproduce are more likely to have affected children. An autosomal dominant pedigree looks like this. So both parents would be affected. Their offspring, half of them might be affected. Heterozygotes are affected, as well as homozygous dominant also. So some examples of homozygous recessive disorders include methemoglobinemia and cystic fibrosis. That's a very well-known condition. This shows you methemoglobinemia and cystic fibrosis, where there's a faulty protein channel and that allows the buildup of mucus. Some autosomal dominant disorders include osteogenesis imperfecta, where you have weakened brittle bones, or hereditary spherocytosis, where the red blood cells are fragile and can burst very easily. Some traits are controlled essentially by more than one allele. That does happen. Most often that happens. Blood type is a really great example of times when you can see like the existence of more than one allele expressed at once. So for human ABO blood types, if you have you have an A antigen on red blood cells, anti-B antibody in the plasma, or B, you can have the B antigen on the red blood cells and anti-A antibody in the plasma, or you can be little I and have neither A or B antigens and both anti-A, anti-B antibodies in the plasma. Phenotypically, that looks like you can have type A blood, type B blood, you can even have type AB, where both are expressed. And if you'll notice, none of those antibodies are in the plasma, because that would be really bad if you had type A and type B and you had antibodies against your own blood type. And here's type O, where you have anti-A and anti-B. You can see codominance sometimes where more than one allele is fully expressed at the same time. And that's like when both A and B are expressed and you have type AB blood. Sometimes you can see incomplete dominance where homozygous red has a red phenotype, homozygous white has a white phenotype, but then heterozygous has a pink phenotype. That does happen. Sometimes you can see incomplete dominance where the dominant allele may not always lead to the dominant phenotype of a heterozygote. So an example of that would be polydactyly, where you have extra digits on the hands, feet, or both. Not everyone who inherits the dominant polydactyly allele will exhibit the trait. You can see an example of pleiotropy, which is what happens is a single gene that's mutated affects 2 or more very distinct, very unrelated seeming traits. Like in Marfan syndrome, you have disproportionately long hands, arms, legs, and feet. And then also at the same time, you have a weakened aorta and poor eyesight. And none of those things really seem like they're, kind of tied together, but they're the result of this one mutation. We talked about autosomal inheritance, but now we have to talk about X-linked inheritance. So for mammals, the X and Y chromosomes determine the sex. On average, females are XX and males are XY. So the term X-linked is used for genes that are carried on the X chromosome. The Y does not carry those genes, and so that can happen often. So if you look at X-linked inheritance, you would see the allele on that X chromosome. You would not see it on the Y chromosome. Some common X-linked disorders in humans include color blindness. It's very common. We see that often. So you can see the blue sensitive protein that's found on the autosomal chromosome 7, but there's also red and green sensitive pigments on the X chromosome. And so if you receive 2 recessive X chromosomes or a recessive X and a Y, that will result in color blindness. Also muscular dystrophy, so you have weakened wasting muscle, hemophilia, the inability to clot blood. This is a really good example of hemophilia in the royal families of Europe. So it shows you issues with hemophilia in their history.